Hello, my name is Sashel King, and this project will be over molecular evolution. Now we'll cover patterns and modes of substitutions. Patterns and modes of substitutions in genetics refer to the ways in which nucleotide substitutions or changes in DNA sequences occur over evolutionary time. These patterns and modes provide insights into the mechanisms of molecular evolution, the rate of genetic change, and the forces that drive genetic diversity within and between species. So let's start with a short introduction. Substitutions involve the replacement of one nucleotide, A, T, C, or G, with another in a DNA or RNA sequence. These substitutions can be silent, or they can result in amino acid changes in the proteins. So let's dive into our types of substitutions. On this slide, we'll talk about transition substitutions and transverse substitutions. Transition substitutions involve the exchange of a purine, A or G, for another purine or pyrimidine, C or T, for another pyrimidine. Transverse substitutions involve exchange of purine for pyrimidine or vice versa. So to continue, we'll talk about patterns of substitution. Patterns of nucleotide substitution describe the frequency and the direction of changes over time. So let's talk about the directional bias. Some substitutions occur more frequently in one direction. For example, A to G is more common than G to A. This can be due to the differences in mutation rates or selective pressures. So now let's move on to transversion bias. Transitions are generally more common than transversions in many genomes. This bias is attributed to the chemical similarity between the bases, purines and pyrimidines, involved in transitions. And lastly on this slide, we'll talk about mutation hotspots. So certain genomic regions or specific nucleotide motifs are more prone to mutations than others. These are referred to as our mutation hotspots. So now we'll talk about modes of substitution. Modes of substitution describes the underlying mechanisms driving nucleotide substitutions. So we'll start with our first mode, neutral substitution. In neutral evolution, most substitutions are selectively neutral, meaning they do not confer a significant advantage or disadvantage to the organism. Neutral substitutions accumulate at a relatively constant rate and are the foundation of the molecular clock hypothesis. Secondly, we'll talk about positive selection, also known as adaptive selection. Positive selection occurs when a new allele provides a fitness advantage and becomes more prevalent in the population. This results in an increased rate of non-synonymous substitutions, amino acid changes. Next, we'll move on to our negative selection, or purifying selection. Negative selection acts to preserve the existing function of a gene or protein. It leads to a reduced rate of non-synonymous substitutions because most mutations are deleterious and eliminated by natural selection. We can also talk about balancing selection. Balancing selection maintains genetic diversity by favoring multiple alleles in a population. This can lead to increased polymorphism and a higher rate of non-synonymous substitutions. So how can we apply these? So understanding patterns and modes of substitution is crucial for various fields, including phylogenetics, population genetics, and molecular epidemiology. It helps in estimating evolutionary relationships, divergence times, and the impact of selection on genetic diversity. So to conclude, patterns and modes of substitution in genetics provides a framework for understanding how DNA sequences change over time. These concepts have profound implications for our understanding understanding of evolution, genetic diversity, and the functional consequences of nucleotide substitutions in genes and genomes. So now we'll start with molecular clocks. A molecular clock is a concept in genetics, evolutionary biology that provides a method for estimating the timing of evolutionary events by examining the rate of genetic changes, typically at the molecular level, such as a DNA or protein sequence. This concept is based on the assumption that mutations accumulate in a relatively constant and clock-like manner over time, which allows researchers to make inferences about when different species diverge from a common ancestor or when specific genetic events occurred. So let's go over the background of molecular clocks. This idea of a molecular clock was first proposed by Emil Zuckerkandl and Linus Pauling in the 1960s. They suggested that the rate of nucleotide substitutions in DNA sequences could be used as a measure of time that has passed since two species shared a common ancestor. The concept of molecular clock builds upon Charles Darwin's theory of evolution, which posits that species evolve gradually over time through the accumulation of genetic changes. So number 2, we'll talk about genetic mutations. The foundation of the molecular clock is the notion that genetic mutations occur randomly and accumulate at a relatively constant rate over generations. These mutations must be synonymous or non-synonymous. The rate of mutation is influenced by several factors, including the type of mutation, the organism's generation time, which is how long it takes to reproduce, and the mutation rate itself. Number 3 would be calibration. To use a molecular clock to estimate the timing of evolutionary events, researchers need to calibrate it. This is typically done by incorporating known historical events or fossil evidence. For example, if the fossil record shows that two species diverged 10 million years ago, and DNA analysis reveals a certain number of genetic differences between them, researchers can estimate a mutation rate per million years. Number 4 would be molecular clock models. There are different models and methods for implementing the molecular clock. The simplest model assumes a constant rate of mutations across all lineages and through time. More complex models may account for variation in mutation rates among lineages or changes in mutation rates over time. Relaxed molecular clocks allow for variation in the rate of mutation among different branches of the evolutionary tree. So on this slide, we'll talk about the applications of molecular clocks and the limitations. So let's start with our applications. Firstly, molecular clocks have been used to estimate the timing of various evolutionary events, such as the divergence of species, the origin of particular genes or traits, and the timing of population expansions or contractions. They have been applied in a wide range of fields, including paleontology, anthropology, and epidemiology. For example, molecular clocks have been used to estimate when humans and chimpanzees last shared a common ancestor and when certain infectious diseases like HIV jumped from animals to humans. So let's move on to limitations. Molecular clocks are based on several assumptions, including a constant mutation rate and a lack of selective pressures that would influence the rate of genetic change. In reality, these assumptions may not always hold true. Calibration can be challenging, especially for lineages with poor fossil records or when historical events are uncertain. Molecular clocks may not be accurate for very short timescales due to fluctuations in mutation rates and the effects of natural selection. So to tie it all together, the molecular clock is a valuable tool in genetics and evolutionary biology for estimating the timing of evolutionary events based on the accumulation of genetic mutations. While it has its limitations, it has provided valuable insights into the history of life on Earth and continues to be refined with advances in genetic sequencing and modeling techniques. Now we'll move on to variation in evolutionary rates. Variation in evolutionary rates is a fundamental concept in genetics and evolutionary bio that refers to the idea that different genes, traits, or lineages can evolve at different rates over time. This variation can occur for a variety of reasons and has significant implications for our understanding of evolutionary processes. Okay, so on this slide, we'll talk about factors contributing to variation in evolutionary rates. Number one, we have genetic elements and regions. Different parts of the genome can evolve at different rates. For example, non-coding regions, or introns, tend to accumulate mutations more rapidly than the coding regions, or exons. Regulatory elements such as enhancers and promoters can also evolve rapidly, influencing gene expression and phenotype. Number two, we have functional constraints. Evolutionary rates are often influenced by a functional constraint. Genes or traits that are crucial for an organism's survival or reproduction are subject to stronger purifying selection, which reduces the rate of change. Conversely, genes with less critical functions may evolve more rapidly due to the reduced selective pressure. And number three, we have adaptive evolution. Some lineages or genes experience rapid evolution due to the adaptive changes that provide a selective advantage. This can lead to bursts of evolutionary innovation. Examples include the rapid evolution of antibiotic resistance in bacteria or the evolution of traits like camouflage in prey species. So let's talk about some more of these factors. So let's start with mutation rates. Variation in mutation rates can lead to a difference in evolutionary rates. Genes with higher mutation rates are more likely to accumulate genetic changes over time. Factors affecting mutation rates include DNA repair mechanisms, environmental factors, and replication fidelity. Number two, population size and demographics. Smaller populations often experience more pronounced genetic drift, which can lead to a faster fixation of mutations and accelerated evolution. Conversely, large populations may experience more efficient purifying selection, slowing down evolutionary rates. Lastly, on this slide, we have selective pressures. The intensity and nature of selective pressures can vary across species and environments, leading to different rates of adaptation. For example, rapidly changing environments may exert strong selective pressures, driving faster evolution. On this slide, we'll talk about implications and we'll add some examples. So, diversification. Variation in evolutionary rates contributes to the diversity of life on Earth. Different lineages can undergo different rates of speciation and diversification, leading to the rich tapestry of species we observe today. So let's move on to human evolution. The human genome exhibits variation in evolutionary rates. Some genes have undergone rapid evolution, contributing to the uniquely human traits that we have, while others are highly conserved due to their critical functions. Next, we have disease evolution. The rapid evolution of pathogens such as viruses and bacteria can lead to the emergence of new diseases and challenges for public health. Then lastly, we have conservation of biology. Understanding variation in evolutionary rates is important for conserving endangered species. Species with slow evolutionary rates may be more vulnerable to environmental changes. Okay, let's go over some of the challenges and what the future holds. So, data availability. Gathering comprehensive genetic data from diverse species and populations is essential to study variation in evolutionary rates effectively. We have our modeling complexity. Developing models that can account for the interplay of multiple factors influencing evolutionary rates is an ongoing challenge in the evolutionary biology. Thirdly, we have emerging technologies. Advances in genomics and computational biology are allowing researchers to study evolutionary rates in greater detail, providing new insights into the mechanisms driving variation in evolution. So, to conclude, variation in evolutionary rates is a fundamental aspect of evolutionary biology. It is shaped by a complex interplay of genetic, environmental, and ecological factors and has profound implications for the diversity of life, human evolution, disease dynamics, and conservation efforts. Understanding this variation enhances our comprehension of how life on Earth has evolved and continues to adapt to changing conditions. So let's move on to a new section, molecular phylogeny. Molecular phylogeny is a powerful field of genetics and evolutionary bio that uses genetic information, typically DNA or protein sequences, to reconstruct the evolutionary relationships and history of organisms. It provides insights into how species are related when they diverge from common ancestors and how they evolved over time. So let's go over a quick intro. Phylogeny is the study of evolutionary history and relationships among organisms. Molecular phylogeny focuses on using molecular data to construct phylogenetic trees or networks. Molecular data such as DNA sequences provide a reliable and objective bias for studying phylogeny because they reflect the genetic changes that occur over evolutionary time. Molecular markers and sequences. Molecular phylogenetics relies on specific molecular markers, such as genes or proteins, that can be sequenced across multiple species. Common markers include ribosomal RNA, mitochondrial DNA, and protein-coding genes. Sequences are aligned to identify homologous positions and analyze the similarities and differences among species. So let's go over our steps involved in molecular phylogenetic analysis. So first, we'd have to collect our data. So we'd collect DNA or protein sequences from a group of species of our interest. These sequences should be carefully chosen to represent a diverse set of taxa. Next, we'd have to sequence alignment. We'd align the sequences ensuring that homologous positions were correctly matched. Gaps would be inserted to account for insertions or deletions. Thirdly, we would construct our phylogenetic tree. So we can use various methods such as maximum likelihood and neighbor joining. These are used to construct phylogenetic trees that represent the evolutionary relationship among the taxa. Then we move on to bootstrap analysis. We'd assess the statistical support for the nodes in the tree through resampling techniques like bootstrapping. Lastly, we'd have tree visualization. We display the resulting phylogenetic tree, typically as a branching diagram, where nodes represent common ancestors and branches represent evolutionary relationships. So molecular phylogenetic analysis can utilize different types of molecular data. Number one, we have our nucleotide sequences. DNA or RNA sequences, including coding regions or non-coding regions. We have our amino acid sequences. Proteins can also be used for phylogenetic analysis, especially when coding regions are conserved. Lastly, we have our genome-wide data. Whole genome sequences provide a comprehensive view of an organism's genetic makeup and can reveal more complex evolutionary relationships. Okay, so this slide covers our applications and challenges. So let's start with species classification. Molecular phylogeny helps classify species into hierarchical categories based on their evolutionary relationships. Now we'll move on to understanding evolution. It provides insights into patterns and processes of evolution, including the timing of divergent events. Comparative genomics. It aids in the idea of conserved genes, functional elements, and genomic innovations. Okay, so let's move on to data quality. This will be one of our challenges. Accurate sequence data are crucial, and errors or biases in sequences can lead to incorrect phylogenetic conclusions. Model assumptions. Phylogenetic methods make assumptions about evolutionary processes, and violations of these assumptions can affect results. Or long branch attraction. Some methods may be prone to artifacts when analyzing distant related taxa with long branches. And lastly, are incomplete lineage sorting. Complex evolutionary histories can lead to incongruences between gene trees and species trees. So to wrap this all up, molecular phylogeny is a valuable tool in genetics and evolutionary bio that provides a detailed understanding of evolutionary relationships among organisms. It has a broad applications in various fields and continues to evolve with advances in sequencing, technology, and computational methods. Molecular phylogeny contributes to our knowledge of the tree of life, the history of life on Earth, and the genetic basis of biodiversity. Okay, our last section covers acquisitions and origins of new functions. So on this slide, we'll go over mutations and genetic variation, gene duplication, and gene fusion. So let's start with mutation and genetic variation. The foundation of acquiring new functions lies in genetic variation. Mutations are the primary source of genetic diversity. We know mutations can occur spontaneously during DNA replication, recombination, or due to environmental factors such as radiation or chemical exposure. We also know that mutations can result in changes to DNA sequences, including single nucleotide changes, insertion, deletions, or rearrangements. Okay, so let's talk about our gene duplication. One of the most common mechanisms for acquiring new functions is gene duplication. This occurs when a gene is accidentally duplicated, resulting in two identical copies. One of the copies can retain the original function, while the other copy becomes free to accumulate mutations and evolve new functions. The original and the duplicate genes can also diverge in function by partitioning or the original gene's functions between them. Lastly, on this slide, we'll talk about gene fusion. Gene fusion occurs when two previously separate genes, often with related functions, become fused into a single gene. This can result in a novel protein with combined or modified functions, potentially providing an adaptive advantage. Okay, on this slide, we'll cover exaptation and co-option, horizontal gene transfer, and evolutionary arms race. So, exaptation and co-option. Exaptation refers to the process in which a pre-existing trait or gene function takes on a new function that was not the result of natural selection for that specific purpose. Co-option involves the adaptation of a gene or trait for a new role without significant genetic change. These processes often involve repurposing existing genetic elements for new functions. Okay, so we'll move on to our HGT. HGT is the transfer of genetic material between unrelated organisms, often through mechanisms like plasmids or viral vectors. This can introduce entirely new genes or functions into an organism's genome, rapidly expanding its genetic repertoire. Lastly, we'll talk about the evolutionary arms race. Organisms can acquire new functions in response to co-evolutionary interactions with other species. For example, in the context of host-pathogen interactions, the host can evolve new immune functions to combat pathogens, while pathogens can develop new mechanisms to evade the host's defenses. Lastly, in acquisitions and origins of new functions, we'll go over experimental evidence, adaptive evolution, and our molecular mechanisms. So first, let's talk about experimental evidence. The acquisition of new functions has been demonstrated in laboratory experiments, particularly with microorganisms like bacteria. Experimental evolution studies have shown how novel functions, such as the ability to metabolize new substrates or resist antibiotics, can arise through mutation and selection. Second, we'll talk about adaptive evolution. The acquisition of new functions is often driven by natural selection. Mutations that result in advantageous traits or functions are more likely to be passed on to future generations. Beneficial mutations can lead to the fixation of new functions in a population, contributing to evolutionary adaptation. Lastly, we'll talk about molecular mechanisms. The acquisition of new functions at the molecular level often involves changes in gene regulation, protein structure, or the interaction between genes and proteins. Understanding these molecular mechanisms is essential for elucidating how new functions emerge. So, to wrap it all up, the acquisition and origins of new functions in genetics are fundamental to the process of evolution. Genetic mutations, gene duplication, gene fusion, exaptation, co-option, HGT, and co-evolutionary interactions are some of the mechanisms through which new functions can arise. These processes underpin the diversity of life on Earth and contribute to the ongoing adaptation and evolution of species.